interaction of etoposid and the multiple carbon nanotube. So first of all, I have prepared something, the introduction of etoposid, but as she already told about etoposid, what is that? But I'm just going to say some few words like etoposid have so this kind of structure it has, and it can be used in several kind of chemotherapeutic agents. In case of chemotherapy, some kind of other chemicals, other medicine can be used, and that is called combo therapy or combination of therapy. So in that case, it is etoposid is generally used for chemotherapy, and we can use it for several kind of cancer. I'm going to show you something. No, it is already told by her. So it can be used by oral medicine and injections also. And in case of injection, etoposid should be put very slowly, because when people are getting etoposid, so the blood pressure is suddenly going down. So we need 4 hours to inject slowly, like that. So these are the cancers. These kind of cancers can be cured by etoposid. But the etoposid, the main mode of action is like it is a replication fork. So etoposid is affecting this enzyme. This is topoisomerase. So we have two kind of topoisomerase, 1 and 2, and in case of when the replication is going on, the DNA, this is helicase. So when helicase is going to separate the strands, so the DNA got some problems, like it's called supercoiling. Supercoiling is like coiling is like something is wrapping something in between, and supercoiling is the coiling of the coiling itself. So topoisomerase, it releases the supercoiling, and if the topoisomerase is not functioning, so the replication can be stopped at any time. So etoposid is kind of topoisomerase inhibitor. So topoisomerase what does? So already people, the other lecturer, they already told that the etoposid it breaks the DNA. How it breaks is like topoisomerase. Its mode of action is like it cut one of the strand of the DNA, and it rejoin to the other side, and this coiling would be released. So topoisomerase is like that, and topoisomerase 1 and 2 is like that. First of all, topoisomerase 1 cut the DNA, and topoisomerase 2 rejoin the DNA in the other side. So it releases its coiling. So what does the etoposid do? Like it stops the topoisomerase 2. So first of all, the DNA cut, but it cannot be rejoined in the other way. So the DNA is in that way DNA got damaged. And in that case, etoposid in that way, etoposid control the cancer cells, because cancer cell. What is the difference between cancer cell and normal cell? That is, in case of cancer cell, it loses its control of cell division. So in normal cell division we have some that is called the halting point or cell cycle checkpoint. So in that case, cells in the metaphase or in case of mitosis or meiosis, so we must have some halt of that cell division. But in case of cancer cell, there is no no haltage. So it's gradually dividing and dividing and dividing. So if etoposid stop the topoisomerase, so it cannot be divided more. So what is the difference? In case of cancer cell, it needs more topoisomerase activity. And if etoposid stop this topoisomerase, so cancer cell will be affected more than the normal cells. But what happens? Etoposid doesn't know which is a normal cell and which is cancer cell. So actually it affects both of these cells. So in case of our body, those cells are rapidly dividing in our body, that is affected more in etoposid in comparison to others. Like our mouth squamous cells, it's rapidly dividing, hair follicles and something. So if we use etoposid, so this kind of the blood cells also is rapidly dividing. So these kind of cells are affected by etoposid. So this kind of the side effects can have when the etoposid chemotherapy is going. So that's why we need more detailed knowledge of how to apply the etoposid so that the side effects and other things can be decreased. So that's why our experiment will explore better way how I mean, without side effect we can have the cancer chemotherapy, and we can have the etoposid to treat the cancer. So we have already Professor mentioned about the single wall and multi-wall carbon nanotube. So we have used this multi-wall carbon nanotube, and we got good results with etoposid interaction. So this is my I mean, very short experimental details. Like, first of all, we used the multi-wall carbon nanotube, and we make solution in a proper way. Then we used etoposid to combine it, and after that we detect by electrochemically differential pulse voltammetry that already I explained to you, and this figure is shown by Y also. But this is the result of that interactions, like in the top. This is only glassy carbon and multi-wall carbon nanotube. And after that, we gradually increased our concentration of etoposid, because we have used different concentrations. So to know which concentration is best for the interaction of the etoposid and carbon nanotube, so we found this is the best. This is 14.4 millimolar. And this is the scheme. I mean, already we explained to you that we can have different kind of interaction with glassy carbon electrode with etoposid. Like, first of all, we can interact with multi-wall carbon nanotube, then etoposid, or etoposid, then multi-wall carbon nanotube. But in that case, firstly we prepared this in this. So after that we used, and we got good results, and this is really good contaminant control. So we found that we are able to interact the etoposid with multi-wall carbon nanotube, and we can go for further experiments. So further experiments is as Professor already told you, that the further experiment will be the interaction of DNA, I mean double-stranded DNA or oligos, or it would be interacted with magnetic nanoparticle. So that the total complex can be taken out easily. And the most important thing is that I think why ask a question that if we use only one of the medicines like etoposid without combination of other medicines, so maybe the effect will be decreased. But in that case, we think that no, because we are planning to use the antisense. So maybe the antisense can have more power, because etoposid only interacts with topoisomerase, and in all the cells, if etoposid attacks the topoisomerase, and and along with this, simultaneously, if the antisense interact other genes to silence it, so it would be more effective, more and more effective than the combination of therapy, I think. Because it can stop the cell cycle of cancerous cell or carcinogenic cells, or both the ways, like interacting with some drug and also in molecular biological way. Because nowadays kind of anti-cancer, antisense therapy is going on. Like we can, some of the genes are really responsible for cancers. As you have know that some carcinogenic oncogenes are several kind of oncogenes, proto-oncogenes that is really responsible for the cancer. Because if we want to know why we are getting cancer, we don't know it still. Some of the reasons we can have, like the smoking or something, the tobacco and some kind of radiation or something, but we don't know actually why. If people don't have any kind of carcinogen, so then also they have the cancer. What is the reason? That is because of some kind of some reason that comes from our heredity, some reason that comes from our DNA or genes or something. So that genes can be stopped by antisense, and it is if we use different kind of antisense in our, like first of all we have planned to use the antisense of breast cancer, and after that, as Etoposid is working on different kind of cancers, so we can use different antisense of different genes. So this project, I think this product will have a various dimension of way so that we can control the cancers in different way, I think. So, and also apart from this, maybe in future we can have some kind of other plannings, like as we have the double-stranded DNA in between our products. So double-stranded DNA also can have the potentiality to interact some kind of proteins, like zinc finger or something. Some proteins can also interact with DNA. So if in future we are able to interact some proteins, that proteins may have some kind of some kind of anti-cancer property, or may have some peptides also in that case. So that would be a new dimension of that kind of products and that kind of medications. So I think in future we can have several dimensions and several way of thinking, so that we can control the cancer in different way, in our way also. Thank you.
Thank you for the presentation. And I am opening discussion. Answering question.
Amitavaa, do you know the liposomal form of etoposид molecular means?
No, still I don't know. I don't have any information. Maybe I can search and confirm, but still I don't have any information. Liposomal function.
One more question is about, do you think that its final concentration of etoposid or capacity of multiple is not, let's say,
Final concentration.
Yeah.
That means no, we are not sure. Maybe some other concentration can be tracked, but right now it's up to this kind of experiment we have this concentration. So
It is, I think it is very important information, because higher concentration inside in this carbon nanoparticles is better, because I think this is carrier or transporter for this drug, and slowly diffusion in cell, and long time, because we linear drip application anti-cancer drug, we obtain only very rapid high concentration this anti-cancer drug, and very rapid decrease this concentration. 1 hour, 2 hours, and this therapeutic concentration is disappear. And this technology open long time, high concentration this drug in cell.
but in our cases, the limitation was, the concentration of etoposid was too low. That is available right now. So, in that case we can we are able to measure this. This was ours. I mean, for the available etoposid, that was the maximum is 14.4 millimolar.
This is also my question, because Professor, now we don't have etoposid anymore.
Yes, it is good concentration.
I mean, future work. We don't have like more etoposid.
Yeah, okay, we can buy. We have to buy. We must buy. Okay, I understand.
because we got from I got from it was 50 millimolar and 15. Okay, maybe no 50. 50. I think
Because you take one microliter, 2, 5, 8, and 9.
yeah.
So maybe 10 to 5.
Okay, whatever. So it is the final concentration of the solution.
Yeah.
So it is the maximum. It is the maximum from that etoposide. And also that information should know everybody. The amount is very low. 12 milliliter, I think.
Yeah.
Not milliliter, microliter.
Yeah, micro.
So in that case, okay. So it is our maximum try.
Okay, other questions? Same shot?
This graph shows that the electrochemical signal depends on the concentration of the molecules. What happens if you have different concentration of carbon nanotubes? Does the signal change somehow?
I think we can answer this.
Okay. I read one similar article with fluorine and etoposид. When we try to increase the concentration of fluorine, the signal decrease because the fluorine work as a it's with separate reduction signal, so it decrease. Our article about the
Fluorines, they don't have to increase the signal at all. So the carbon nanotubes, they increase the signal.
One thing I would like to inform you that in all the reactions, the concentration of the carbon nanotube was the same.
So you will never try different.
No, that is different issue. But in that case, we try to know what is the interaction between carbon nanotube and the etoposide. Okay. So here, all the samples, the concentration of carbon nanotube was the same. So the change of the signal only signifies the interaction of the etoposide, not.
Yeah, and the question is about what happens when we increase the concentration of multiple carbon nanotubes. What will happen? Yeah, and so similarly, I read one article, our article, the decrease signal.
Other questions? Okay.
And
Okay.
Amitavaa, you said by using the design, we can increase the ability of treatment. But you know why we have to combine protein and etoposид to increase the signal. I mean the molecular biology.
No, I'm not understanding you.
Okay, so we usually have to combine protein and etoposид. You know why? Why we have to combine this?
Combination of therapy that is going on recently. I mean, the doctors, they always put the cisplatin and etoposид to get together. Why it is?
Yeah.
Okay, it is some kind of combination therapy is always going on in case of very critical systems, not only for cancer treatment. It can be also for tuberculosis treatment also. Combination therapy, the principle of the combination therapy is like that. Everybody cannot respond to the same medicine in the same way. Like etoposид can be good for you, or cisplatin may be good for me. Who knows? Because every cell have its different conformation, has its different chemical way of chemical environment, better to say. Okay, and in case of cancer, there are several kind of cancers in our body. So each of the cancer should be treated in different way. So it's a kind of increasing the possibility. It's nothing like that. Okay, only etoposид can cure the cancer. The problem is that etoposид cannot cure all the cancers. And doctors, they not 100% sure that which cancer should be used in which kind of medicines. So that's why sometimes they use a combination of therapy, and eventually it increases the possibility of the treatment. Eventually it happens, because those techniques that we have already to detect the cancer, that is not sufficient. That's why throughout the world the scientists are going to involve in cancer research. So that's why we're not okay. We know that okay, this cancer may be from outside, from our intestine, maybe some other disease. Okay, 90% of the possibility that it comes from some kind of maybe some colors or something, some toxicological things, maybe something, and maybe 10% reason maybe it comes from some genetic factors. So we are not sure. So that's why always combination of therapy is good. And another thing is that if you want to cure the cancer with only etoposид, so you need to apply more concentration. And if you want the combination of therapy, each of the medicine should not have the more concentration, maybe 50-50 concentration. So toxicity may be decreased in that way. So combination of therapy is a good thinking of getting the higher possibility of treating any of the diseases. This is the basis of the medication.
Okay. So it means if we want to use only etoposид, I mean we use something like just water, so we should know the body of people is appropriate for etoposид or not first, right?
Yeah.
Because we usually use both, two or three, to know which reaction, because we are not sure. So, but now if we use only one etoposид, so
Yeah.
we should know this works in what body first.
Yeah.
Okay, other questions? Okay. I am very happy because this seminar was nice, and we discussed about this technology, about this thinking, about this process, and other way for this experiment and technology. Thank you for your presentation, for discussion, and I finished.